we pooled all the resources and got 611 people randomly assigned to try low-fat or low-carb for 12 months. It took 3 years to recruit the 611 people, put them through 12 months of advice and counseling to try to eat this way. And originally we had a pretty ambitious data collection set up so that we were getting diet records from them, we were getting blood samples from them. And in the end, they lost 6,500 pounds. And equally, on average for both groups, they both lost about 13 pounds on average after the year. In both groups, somebody lost 50 or 60 pounds and somebody gained 20 pounds and everything in between. So just as we expected, we didn't get much average difference, but this huge variation within. And now we've started to test what are the explanations for this. And as we were originally archiving samples for things like DNA, right at the beginning of the study, I got an email from an uppity woman in Europe who said, I got this idea. I think we could make more of your study if I came and helped you and did a postdoc at Stanford. And it was so compelling that we graciously and gratefully invited you to come join us, where you worked with Robert Hale and Megan Hitchens, and have brought an entirely new dimension to the whole study. So whereas we had this idea that there was a genetic predisposition to weight loss, which actually didn't work out. We've already done the test. The genetic predisposition didn't work. But because we collected DNA, it created the opportunity to look at the methylation or the epigenetics. So let's swap. And now it's your turn to talk a little about how epigenetics would work in this study.
I've been working in epigenetics now for 10 years. When I wrote that email to you, I was in Oxford. I remember that night. And at that time, I was working in epigenetics with simple model systems, yeast cells, ciliates. And I really wanted to study the link between epigenetics and nutrition. And I'm glad that I wrote you that email. At that time, personalized medicine, the field of personalized medicine was growing. So personalized medicine is a new healthcare approach that tailors treatments like a drug, but also a diet intervention to people's unique characteristics.
It makes so much sense. So when they get drugs and drug doses, there isn't one drug that works for everyone at one dose. Why would there be one dietary approach that works for everyone? So you came just in the nick of time, Lucia.
Exactly. And the basis of personalized medicine are genes, microbiome, and epigenetics. And we are working on all these three aspects in our study now, which is called Dietfits.
Dietary interventions examining the factors interacting with treatment success.
Wow.
I love it because it seems to fit our study. We were trying to see if there was one diet that would fit better with one individual than another because of their genetic, microbiotic, or epigenetic predisposition.
Yeah, exactly. And the epigenetics part, I think, is very important. So let's explain first what is epigenetics. The Greek prefix epi means on the top. So literally, epigenetics means on the top of genetics. And physically, on the top of our genes, there is a second genome, where the genome is your DNA. So a second genome that is called epigenome. And it is made of molecular switches that can turn genes on and off. Just like a dimmer switch can modulate lights up and down in a room. And these switches are called epigenetic marks. And the process of the epigenome of turning genes on and off is called gene expression. And gene expression explains why, for example, we have different cells in our body. Liver cells, brain cells, fat cells, all told 300 different type of cells, all sharing the same DNA.
Right, they all got the same code down there. So how come they're not all producing eye cells? Because of the light switches.
Exactly.
Okay.
You can think of your genome as the hardware, and the epigenome is a software that tells your DNA what to do so that different cells can form. And this brings us to an important property of epigenetic marks called epigenetic memory. I don't know if you remember, in 2010, the Time magazine dedicated an entire issue to epigenetics. And the cover story? Why your DNA isn't your destiny. How the choices you make can change your genes and those of your kids. You remember that?
Which is good, because we don't want to be fated to ill health if it looks like we're predisposed. We want to save ourselves.
Exactly. And the reason behind this is that we cannot change our genes, but we can change our epigenetic marks because they are dynamically modified by our lifestyle choices. Things like diet, exercise, stress. So this bring us back to our opening topic. So what makes us unique? Our genes, but also our lifestyle that can change our gene expression through epigenetic marks. So we are unique in two ways. Because of our genome, the book we are born with, and because of our epigenome, the book we are the author of.
I like that analogy.
I like that too. One of the best studied factors affecting epigenetic marks is diet.
Oh, I love that.
Yeah. And diet intervention studies are an ideal framework to test whether diet-induced epigenetic marks are reversible after weight loss. There are epigenetic marks that are specific of overweight people, and other epigenetic marks are specific of lean people. And when overweight people lose weight, their epigenetic marks become more similar to those specific of lean people. And one of these studies even showed that these epigenetic marks in overweight people are found in sperm cells.
Oh my gosh.
Yeah, so these changes can be potentially passed from one generation to the next, just like the cover story of Time magazine. And but there is a problem with all these studies of weight loss and epigenetics. Most of them are were done in small populations and for a short duration. And our study with 600 people and 1 year intervention is the largest and longest of this kind. And we want to do, we are looking at two aspects of epigenetic marks. First of all, whether epigenetic marks of overweight people can change after weight loss. But also whether there are some epigenetic fingerprints specific of different people that can predict weight loss outcome after a diet intervention. And these epigenetic fingerprints are called epigenetic biomarkers. We have heard of genetic biomarkers. They can predict disease risk or for example, diabetes or cancer, also the response of people to drugs or treatments. And for example, our genetic marks, the genetic marks that you identified in your study that could potentially predict the response to a low carb or a low fat diet. Epigenetic biomarkers can do the same thing, predict disease risk or treatment response, but have an advantage over genetic markers. Because you cannot change your genes, then genetic markers can tell you only what you were born with, where your starting line was. Whereas epigenetic markers can tell you where you are now in your journey to optimal health.
What do you do when you go in the lab and look at this stuff? Because it actually boggles my imagination how you try to focus in on some component or area of the DNA.
Yeah, you're right. The DNA is pretty long. You know how long it is?
I'm not even going to guess. Sorry, I'll just embarrass myself.
Is around 2 meters long, so 6 feet.
Uh-huh.
Yeah. And so can you guess how we can possibly pack it in 2 micron, which is 2 millionth of a meter, which is the space where the DNA sits in the cell?
Tightly coiled.
Wow, you're prepared. Yeah, tightly
coiled. It's like my phone cord, tightly coiled.
Yeah, just a phone cord. And you know how the phone cord of your cells is called?
The double helix.
Double helix plus protein histones, which makes the chromatin.
Okay.
So chromatin is a very important concept in epigenetics. It's what the DNA sequence is in genetics. So it's a way of storing information. If you think about it, our DNA sequence stores the information, our genetic information, and our chromatin stores our epigenetic information. The point is that the DNA is so long, so we have to pack it around histones and make it very compact in our cells in the form of chromatin. Now, in this form, we cannot read the genes. We cannot read the sequence, and we cannot make protein out of DNA. So what we have to do is to open the chromatin to read the genes and close it back to turn genes off.
Okay.
And this is what epigenetic marks do. They open the chromatin and
allowing expression. Okay.
Yeah, turn genes on and close the chromatin and turn genes off. And these are what all these switches do.
So wait, let me ask a follow-up question. So epigenetics isn't required to open the chromatin. It's just one way to open the chromatin. Is that a fair question?
Yeah, it is. Okay. Yeah, so it's one way of opening the chromatin. There are transcription factors, which are basically gene reading machineries in our cells that do that. But epigenetic, so the difference between epigenetic mechanism and the other gene regulation mechanisms is that epigenetic mechanism can be inherited through cell division. So, and this is the point. So, you know, when one cell divides, it duplicates the DNA and then gives rise to daughter cells. Now, the epigenetic mechanism can be copied to the daughter cells, so they are stable through cell division. Whereas the other mechanism of gene regulations that are regulated by other enzymes or protein are not copied to the daughter cells. So this is what makes epigenetic mechanisms different.
Okay, thanks. All right, so where are we at with this study? So we've got this great study. We got all kinds of archived blood samples, And there was a fabulous paper that came out just a couple of months ago that maybe led us in a new direction. I remember you being excited about this paper.
Yeah, this was a very exciting study. It was a longitudinal study, not an intervention study.
So they need us to follow up, don't they? Okay,
they
good.
need us.
We're here for
them.
them.
They need us.
They need you.
Yeah, this was so the study was published in Nature in January 2017. And shows, they identified 62 epigenetic markers that can predict the risk of developing type 2 diabetes independently from common risk factors like insulin, blood glucose, and body mass. And the top candidates are genes that are very interesting, have some biological functions in regulation of insulin secretion. And so we want to find out if we can replicate these markers in the context of an intervention study, and whether these markers are reversible. We started with a very costly technique, which is called genome-wide bisulfate sequencing, which basically looks at the epigenetic marks throughout your DNA, the junk and the genes.
That's more than one or two.
Yeah.
Okay.
A lot.
Okay.
And this experiment, as you know, is can cost up to $8,000 per sample.
Yikes.
So we did a deep dive on a selected sample. But now we are actually testing the top candidate genes in all the people from the studies using a cheaper approach. This I think costs about $30 per sample. It's called pyrosequencing. Okay. And basically with this technique you can just analyze the epigenetic marks as a specific gene of interest. Can tell you where you are now in your journey to optimal health.